So, guys, I think we can start. Good morning. Good morning, Jeff, Ari. Thanks for joining me.
How's it going?
Good morning, George.
Good, good. So, just to introduce you to our audience, to Science Me audience, we have together Jeff Stubbs, Environment and Food Safety Senior Business Development Manager for EMEA from THERMO Fisher Scientific. And we have also Aristide Ganci, Product Manager, Environment and Food Safety Business Segment for THERMO Fisher Scientific. So, welcome, guys. Thank you very much for being here.
Thanks for the invite.
Yeah, thank you.
So, this is the second row of our episode for PFAS, and in the previous we discussed mainly about Orbitrap, okay, and actually the hardware of the whole thing. And now we are going to, let's say, give a step back about how we prepare the sample before introducing this to the Orbitrap, right? So we're going to talk about sample preparation. And to be honest, to my perspective, there is a minority of people that talking about sample preparation. I mean, not from you guys, but you know what I mean, right? Everyone is talking about the hardware. I would need probably an Orbitrap or TOF or triple quadruple or whatever, but no one talks about sample preparation. So I would need a comment from both of you because I'm aware that THERMO Fisher Scientific has a complete bunch of portfolio about sample preparation, right? Any comment about that?
Yeah, I'll go first and maybe give a bit more of an overall overview. I worked for a long time in a lab analyzing a variety of water samples, contaminants, pesticides, and my opinion is that regardless of the instrumentation that we use in these labs, and there's some fantastic equipment out there these days, a lot of it's down to the sample prep. One of my good customers one time said, if it's rubbish in, it's rubbish out, and the instrument, whether it be a mass spec, a triple quad, an Orbitrap, whatever, it won't care. It will just analyze what's actually there, and that's why sample prep is so important for me. And also, when we are talking about the routine labs that day after day have to do this, they have to do the same routine from sample prep to analysis to reporting, and it all starts again. That becomes really important to get the sample prep right. If it means that getting it wrong, you have to repeat the analysis because water companies can't let the water out, or are we sure we are within the right limits, etc., etc. And to me, sample prep is the most time consuming and error prone and expensive part of analytical chemistry. So here at Thermo, in the chromatography and mass spec division, we really do try hard to fit some of our technology into an application that is where the sample prep is required. And I think a lot of people forget about this. But when we talk about just a generic auto sampler on your GCMS, your LCMS, whatever it is, it does something that's automated already. And it's the kind of last step of sample prep, which is the injection, because gone are the days where you are standing next to your instrument, ready to, with your syringe, to go put it into there. You've got an auto sampler that does that. And if we can think about that being something that we wouldn't ever want to do, because we've got lots of samples, because we need to spend something else. If you step back into that process, what was before the sample ended up in the vial? What was before that? And if you go all the way back, you start with the bottle or the sample. And that is where I think we need to focus upon. And that's certainly something that we're doing at the moment in my role in the business development. Whilst we want to talk about and really leverage the technology that we have in the analytical side of things, the sample prep is a big thing as well. And like you said, there's not enough people talking about it, and we need to talk about it a lot more. And certainly for things like PFAS, we're finding this is really, really a big topic to discuss and to talk about.
Great. And you, Ari, any comment
Yeah.
about that?
Yeah, I completely agree with what Jeff said, and he covered already a lot of points. I mean, there's only very few matrices where you don't need sample prep, which is basically clean drinking water. But other than that, if your matrix gets more complex, and especially in the environmental and food safety field, we have many different types of matrices, many different types of chemistries, possible interferences. There can be pigments, there can be fatty, oily substances. So sample prep is really the core, as Jeff said, of the analysis, of making sure that you get what you want from, and you find your compounds of interest, and they're not masked behind a huge matrix interference. And that's what we also try to educate our customers to, especially some of them who are not familiar with sample prep. It's an important part of our training we give to customers, that sample prep is a crucial part to the entire workflow.
So, all right, by now we are addressing all the challenges, I think, from what you said, right? So, how does Thermo Fisher, I mean, workflow, portfolio, whatever, address those challenges?
Well, one of the first things they'll do, regardless of the challenge, is speak to their customers and find out what the customer's challenge is, and then translate that into trying to, I guess, position the right technology into an overall workflow that goes from, bottle to report. Of course, there's the nice mass spec or whatever bit, but that is irrelevant if you don't get that sample into the right format to be an extract. So Thermo has a lot of different sample prep techniques and capabilities, from automated solid phase extraction, automated liquid extraction, the right SPE cartridges, a lot of cleanup tools and this sort of thing. But what we're seeing at the moment is, whilst all those are very good and usable, we're seeing a very big upturn in automated sample prep rather than the manual, because people are challenged with staffing these days. Staffing costs, a lot of money.
And a lot of samples again.
A lot of samples, exactly. So actually, you can't pay somebody to be there overnight. They've got to go home and rest to start again. So this robotic automated sample prep, anything that can be done there to be more robust and more easier for, I guess, day to day is certainly something, and we're trying to push into that. And early stages seem to be very, very positive from what our customers are trying to say and tell us.
Okay. So Ari, any comment about that?
Yeah, what I would like to add is maybe that when we talk about PFAS, obviously there's three main challenges when it comes to sample prep. It's obviously we have to go really low detection limits. So obviously the instrument has great performances, but we need to ensure that we get high concentration factors across our sample preparation. Secondly, we need to deal with, as I mentioned before already, many types of different matrices. So cover a whole range of methods which can deal with both environmental and food types of matrices. And contamination is obviously probably the biggest topic for PFAS. So ensuring that we don't contaminate or have no external contaminations in our sample prep when we deal with our samples.
So, okay, thank you, Ari, for the pass. I will take that. I mean, background contamination is one of the key challenges, right? I mean, especially when we're talking about difficult matrices. So, how do we ensure that we avoid any false positives, and how Thermo help the lab to with any kind of solution and consumables to mitigate this?
Ari, do you want to take that?
Yeah, sure, can start with that. Yeah. So obviously, there's the first step, which is consumables, as you mentioned, George. So we as Thermo Fisher Scientific have a whole range of specifically designed consumables which we use for our PFAS analysis. So these go from vials to caps to syringes. And so on all of these, we try to reduce contaminations to a minimum by using specific materials, Teflon-free materials, these kind of things. So all of these are available, and we give our customers a whole list of what consumables to use for their analysis. And obviously, when we analyze things, we always measure some kind of blanks, and to ensure that on each day the blanks and contaminations are down to a minimum. And obviously, that's more on the instrument part, but we ensure that any contamination which is still present is delayed and is separated from our analytical compounds within the actual analysis. We use a delay column to do that, but that's more on the instrumental side of things.
Okay. Jeff, any comment on this?
Yeah, I think from a customer perspective, and you know, that's that's really key to mention here, is that a lot of our customers don't have the time to test this consumable or that consumable or this column, or you know, it's just not how it used to be. You know, everybody needs something to be up and running immediately. So whilst we like testing different columns and challenging ourselves and, you know, these kind of things, when we look from a routine thing where this instrument or this workflow needs to make money or produce reports and meaningful data as quickly as possible, you know, a lot of customers are looking towards the supplier now to kind of do the hard work for them. And what we're doing, you know, our approach at the moment, especially with environmental and food safety labs, is if we get a good enough handle on what's needed, you know, to go from A to Z, if you like, and we know the challenges that are associated with some parts of that, if we can overcome them and say, this is how you would do it, and give our customers like the SOP, the right way to do this, this is a big thing for customers where they kind of trust the process, but only would do that with a large amount of data to show. So for us to have to show them exactly, look, we went through this was a challenge, we changed this overcome the challenge, and now you can see the data and you can see the reproducibility and the quality of data, not just on one lucky injection, on a large batch, because that's how you would need to simulate this as if it was in a lab.
Right.
So what I'm saying is that there's such a different myriad of different consumables and moving parts that customers can try. We've tried to do the hard work by selecting the right ones for that particular application, and then go with them and prove that those are the right things so that the customer doesn't have to. And hopefully that adds value to the discussion that we have with them, and we start talking to them about a bigger project.
Great. It's what you guys clearly described is that we need an ally for the lab analyst. So someone that stands with him and not against him, or in front of him, or in the back of him. So it's clear a message from you. So while we're talking about different matrices, let's say wastewater, human biomonitoring, how the sample preparation strategies differentiate with each other, and what the analysts should know to get the best recovery and accuracy. Mm-hmm. What's your advice or your proposal on that?
You want to take this, Ari? Or?
Yeah, I can start with that one. Yeah. So similar to what Jeff was already saying for the consumable part, the same goes a bit for the sample prep part. So what we try to do is to qualify all of our sample preparation in-house here in our laboratory before we hand over the SOP to our customers. So an SOP will be very detailed, covering specific matrices. And so the hard work has really been done. And what we use is often we use certified reference materials, or we use proficiency tests to ensure that the data quality we get and our sample prep is actually responding to regulations also, and we get the values we need to get. And that being said, we add obviously labeled internal standards to ensure that we get the right recovery and accuracy for all of the different PFAS. So we have, I think at the moment, around 20 labeled compounds just to cover the entire chemistries and polarities of PFAS, which are very different, and which might also behave very different in different types of matrices. And when we qualify methods, an important point also to mention is that we do different approaches. So sometimes we do an extracted calibration, so where we have different calibration points which we extract. But if we have challenging matrices, sometimes we also do what we call matrix matched calibration curves or standard addition. So we add a defined amount of standard to our compound before extraction and see how much we find after at different concentration. And that gives us kind of added confidence that we actually get the results in different types of matrices, and some are more challenging than others. So yeah, so these are all steps we do to ensure that then the customer gets the right protocol.
Great. And you, Jeff, any?
Yeah, I think it's interesting for me, because when I was back in the day working in the labs, I was working with pretty much clean samples, drinking water most of the time. And from a sample prep procedure, sometimes that was put the sample in a 2 ml vial, add some internal standard, and that's it. You try and do that with anything dirtier. Yes, it's an easier sample prep, but you then lose the time somewhere else. So this is really important, for analysts to understand and recognize, is that the matrix of the sample can really cause you problems if you don't treat it, or don't treat it at all, or don't treat it the right way. So for example, whilst we'd love to put a lot of sample onto an instrument, if there's matrix involved, it will probably dirty up the instrument and cause problems, which then starts affecting your recovery and your accuracy. If your accuracy starts called into question, you're going to need to do some maintenance. And you could end up spending a lot of time with an engineer or with you cleaning the inside of a very expensive instrument,
right?
Not running any samples. So you kind of have to then look at, well, what would protect my instrument the most without losing the fidelity of the data. And that's when, cleaning up the sample might be needed. So the mass spec might be sensitive enough not to enrich or need to enrich. So maybe you need to clean the sample up by doing some quick solid phase extraction. And that's fine. But that will protect you've then got to go through the right SPE cartridge to get, to load the right compounds on at the right time and to unload them so they all come off. And that might help. But of course, the dirtier that you get in the sample, the more that SPE cartridge will foul up, it will get stuck. So you've got to go to some kind of liquid extraction again, solid liquid extraction. And what I would say is, for PFAS or many other compounds, the more sample prep steps that you put in, the more uncertainty of measurement and the more error that, especially if it's manual, can be brought in. So if you've got one step, you've only got to get one step right. If you've got to do 10 steps to make sure that the investment of your instrument is protected by having a nice clean extract, you've got 10 places to get that wrong. But also, as we know, the turnover of staff in many of these routine labs is quite quick. That training somebody to get really good at something, you can do that, and then what happens is that person gets promoted or leaves, and then you've got to do it all again. And then you might have had like 6 months of good data, and then you get this newbie that's not as good as doing it as somebody else, and you start getting that error, or they've missed an internal standard. I've been that guy. I've been that guy that's missed internal standards from 100 samples and be told, where's the internal standard? Oh, sorry, I forgot. So we've always got to think smartly about it and really consider the sample prep, because, as I said right at the start, the mass spec will do the job if you get it into the right extraction, into the right extract. So here at Thermo, we're very, very much trying to think about that more, as we said, but also try and standardize a little bit on the same extraction capabilities if we can, to help customers, so that if they've got to do lots of different extractions for different matrices or different compounds, if we can get a kind of universal style, it's easier to be trained, it's easier to be understanding, everybody knows how it works. That kind of helps lab life over a course of a day, a week, a month, a year, and that's massively valuable to what our customers are telling us.
Yeah, I mean, it's clear that sample preparation with sampling, combine those let's say segments, they have more than 80% of the error of the total analysis, all right, in terms of the quality assurance. So it's very important. And I was thinking about, you said that you missed all the internal standards as a new guy or whatever, but so the automation is the solution for that. And how does the thermo sample preparation workflow integrate automation to improve reproducibility and throughput in high demand of those labs?
Maybe I'll go there, and then Ari, you can maybe introduce
Sure.
this kind of platform that we've standardized on. If you look at all the things that you have in a lab, a normal lab, you've got a fume hood, and inside that fume hood there are probably extraction manifolds with SPE cartridges. There are probably flasks, shaken flasks, separating funnels. There are probably centrifuges, vortexes, all these evaporation systems, all these things that will be very commonplace in a routine lab. And normally there will be lots and lots of fume hoods with lots and lots of people going in and out and around. With the advent of instruments becoming more and more sensitive, our representative sample size can reduce, because we can win that back in sensitivity on the mass spec end, if we're talking about mass specs. And what you do is you reach a tipping point where actually the sample size is so small, you can automate it very, very easily by employing some of the sample prep technologies on our auto samplers, which aren't just auto samplers anymore. These are extraction labs on a rail, on a rail-based auto sampler. So if we look at the different things that a lab has in its lab that an analyst would use every day to do their preparation, if we can have those objects, minimize them and have them on a rail and start there, that's a very, very interesting proposition. And we do lots of different things on our rails, just derivatizing or heating or cooling or shaking or whatever. So there's lots of different sample prep steps that can be automated, because now we've got the technology on the auto sampler. But what we've seen, I guess the biggest game changer that we've seen is a technique called dispersive liquid micro extraction.
Right, what we see also in the live from the lab, right, from Mars.
Yeah, the live from the lab. And this is a very much this is a technique that is all fully automated, doesn't need the skill that you would need to do that method manually as well. Robots are going to do it, and they're going to do it today and tomorrow. They don't ask for a day off. They work on weekends, they work on through the night. And these advantages are really good advantages to really employ in routine labs, where it's really important to get everything right first time. So I'm going to hand over to Ari so he can explain the dispersive process, because I think it's a very good thing to discuss on this podcast, because it fuses really good sample prep with automation, and then what we're trying to get out of it at the other end, which is very, very impactful right now.
Right, Ari. I mean, yes, we need that to picturize for us this technique. Yeah.
Sure. Yeah, and I'll explain it a bit without going too much into detail on technical, but just highlighting how it worked, how the principle works. So the only manual step is what we do is we have to fill a vial with our sample, which could be a water, some type of groundwater, wastewater. But we've also, with this THERMO automated sample preparation workflow, we've also worked with solids, some soil samples. We've tried this on food. So we're trying, especially in collaboration with some of our customers who are providing us real-world samples. We're running them through this workflow to test, and sometimes even test them on quite tricky samples. We recently worked on an AFFF, which is an aqueous film-forming foam, which firefighters use to extinguish fires. And these are really full of PFAS, so that's really a challenging sample. So so again, that's really the only manual step to fill the vial. And then from there on, we place it on the auto sampler of the automated rail. And that's when the process starts. So the first step is really the addition of what we call a dispersant and an extractant. Once we add these and we vortex it, we get a cloudy solution, which is really showing that we have a high surface area because of these micro bubbles which are formed. So we kind of extract our compounds of interest into let's say, a quite low volume of extractant. And the dispersant is aiding in this process. So that's what makes it so efficient. And what we end up is we have our sample, and on top we have our mixture of dispersant and extractant. And we take a syringe, or the auto sampler does all automated, obviously, but he takes the upper phase, transfers it to a second vial where we do a second extraction. This is done just to increase the concentration factor, and at the same time also clean up the sample even more. And what happens then is, again, just a liquid extraction. We again reduce the volume, we increase the concentration, and we finally end up with a few microliters at the bottom of the vial. It's a special high recovery vial. And from there on, we take the vial, we place it on the instrument, and the instrument injects the final extract into the instrument, into the analysis. And all of this has a few, I mean, this is a development we've been doing for more than the last year, I would say. And we had in mind a few things, obviously reproducibility and robustness, reducing contamination. So the system, as we mentioned earlier, is using consumables and syringes which are PFAS free.
Mm-hmm.
What we tried also, which is important for more and more labs, is the environmental aspect. So we reduced the solvents we use. We use maybe 2 milliliters of solvents overall for the overall process per sample.
That's great.
And and so that's an important aspect. And obviously time, as Jeff mentioned, if you do some processes manually, especially SPE, can be really tedious. So just to give you an idea, we work with 2 samples at a time. So we do kind of parallel processing of samples in the automated part. And we are around 20 minutes for 2 samples for the entire extraction. So about 10 minutes per sample. And then,
that's great. Great.
So time saving, that's was another important aspect after speaking to many of our customers who say that, they only get through 10 or 12 samples a day, while we, with our solution, we definitely increase the sample throughput.
And Ari, are there any stats that you can share with us about reproducibility or the extraction level of this? technique?
Yeah, so when we qualify our methods, we always try to get the recoveries within the regulatory. There's certain guidelines for these analytical methods, and we try to fall within the, let's say, what is it, 70 to 130% recovery, and accuracy is always below 20% for these kind of extractions. And again, by adding internal standards, that's really how we ensure that we get down to these low numbers. And obviously, sensitivity, as I say, concentration factor is around 200. So we can go to really challenging levels, even for regulations which need these low levels.
Great. I mean, we need to put a link, below from the episode in YouTube and Spotify again to watch this live from the lab episode, because I think it's great for everyone to visualize the whole automation level that you described. So, I mean, while we're finalizing our episode, and let's stick with PFAS. Okay, so this ever-growing micropollutant family, PFAS, is growing, and is growing either that we are discovering or exploring new PFASs, or the regulations, giving us more instructions to watch out. So, is there anything that the sample preparation needs to involve, to the future of these challenges? I mean, what are your thoughts about that?
I'll go there, if that's all right. So we know that PFAS is everywhere. I don't think anybody these days doesn't need to be told that. And like you said, it's from a few compounds to thousands and thousands now. And we can kind of see where the regulations are going, just even in Europe, in drinking water. The numbers are going up. The limits of detection potentially are going down. So hand in hand with more sensitive and more accurate mass spectrometry to detect these PFAS. We've also got to look at the types of PFAS compounds that we're looking at. We've got short chain, we've got long chain, we've got small molecules, big molecules. And we've got to look at things like the volatile PFAS as well, that might not necessarily go through, the LCMS kind of way. So I see potentially two routes here where we're going to need to deal with the volatile compounds, and then the less volatile, I'll call them, as well. But I still think we're going to need universal methods, that because we kind of don't know what we're exactly looking for right now. The more steps that you put in to selectively extract or enrich, you may be losing some of the ones. So very much a non-selective sample prep, a bit like what DILME is, or even a variety of DILME where we can slightly change the conditions of DILME to bring out certain compounds, and then maybe run it again automatically, but with a slightly different condition, different pHs or whatever, combine the extract and then run that, or have something that we can take the extract and use it for GC analysis and LC analysis. That's kind of something that we're looking at. So that's the first thing for me. So I think sample prep must go hand in hand with the technology that we have to analyzed. As samples get more and more difficult in terms of, as Ari was saying, we've gone from water to wastewater to, foams, soils, chicken, egg, everything in the supply chain, everything that's out in the environment. We don't know what we're going to come up with, but something that can deal with all of that, I think, is going to be very, very important, as quickly and as efficiently as it can. So that's on the manufacturers like us to help to develop that, but also on our customers to tell us what they're hearing and seeing as well. The big one for me at some point is, and I was involved in this a long time ago, I think that instead of these labs, having these centralized labs running these samples where they get a spot sample, and the sample gets taken to the lab, and then the lab has to analyze that within 5 days, 10 days, and then the result gets reported. online monitoring, I think, is going to come back. That's where I personally think, and I used to use this. I used to do this for herbicides, and that means where we've got these maybe miniaturized instruments and miniaturized systems that can fit on a water treatment works, or on an industrial effluent, to really keep in check the water at that particular time, and then using this big data or AI to start monitoring more inputs to work out what the outputs might be. I do really think that that's something as well, but the only way that is able to be done is if you've got something that can be automated and taken care of, because you would never get an analyst prepping samples every day for a water treatment works when there are thousands of them across the country. But if you could deploy something that was done where the whole prep was automated, that's where I think we can go. Or on the other hand, we go really, really crazy and we start doing things where the sample is difficult to analyze, it is difficult to extract, but the instruments are that good, they don't need to do the extraction. They're like amazing instruments. Doesn't matter how dirty the sample is, it will analyze the sample down to a really good high-grade data level of data. But that's way in the future.
Okay, yeah, but many times maybe we are aware of the future, but very close to it. So
we never know.
It happens. So Ari, what are your thoughts? What we need to involve in that?
You mentioned quite a lot of aspects. Something we also are working on is using this automated platform for not only PFAS but also other contaminants our customers are interested in. A lot of labs do analyze PFAS, but at the same time they might be interested in doing let's say pesticides, why not polar pesticides and other contaminants. So what we are trying to do is, on the same principle of automated sample prep, is finding protocols which also extract other compounds our customers could be interested in, so they can use that one platform for different types of analysis and extractions. And just coming back to one point which Jeff mentioned already, is the untargeted analysis. Obviously, ensuring that if you do high throughput screening or untargeted analysis, linking also back to what we said in the first episode with the high resolution accurate mass instruments we have. Obviously, we need to ensure that we are not too selective and exclude any potential PFAS which are not regulated yet but are upcoming while we extract our samples. So and we have done some works on this with customers, and the DILMI seems to work really fine also for doing untargeted analysis. And obviously then comes into play software and obviously data analysis of untargeted analysis. But I think that's an important aspect as well.
Yeah. Love your ideas, guys, and your future perspective. So I would like personally to thank you very much for helping me today to discuss this very critical step. I think you understand what I'm talking, right? I mean, you are better experienced than me. So the sample preparation, as I told you, with the sampling itself, no matter what hardware you have, no matter what software you have, no matter how experienced you may be, those two steps are more than 80% of the error biased in the whole analysis. So thank you very much for helping raise this agenda. And we will put the link from the Live from the Lab that you are in with Jeff. You introduced the DLL sample preparation. So thank you very much, guys.
Yeah, thanks for having us on again, George. Been fun.
Yeah, thank you, George. Yeah.